I wanted to finish off on that first one, which is again talking about the concept of, it's another way of thinking about that dynamics of disease. And it's, for most organisms, but particularly in this instance, most living organisms, they have life cycles. You're well aware of life cycles. But we can also think of things like disease cycles, because you're talking in this instance at least two biological entities that have their own individual life cycles, but their interactions leading to disease can be described as a generic disease cycle. And understanding the processes and the timing of that, once again, can be important for formulating effective management. I can kind of get rid of this now. It might make it a bit easier. Whoa, I should make that big. So here's a generic life cycle, an infection cycle where you start with your pathogen and it sits in the soil or sits somewhere else, resides between crops somewhere, produces an infective agent here. Might be a spore if it's a fungus, it might be it's a cyst nematode and it germinates and a little nematode starts swimming away. Whatever. So the thing that actually is infective, the infective agent gets produced. It comes into contact with the plant itself, which we call inoculation. So the inoculum is the pathogen spore, the pathogen entity. Inoculation is the transfer of that to the plant itself. So it attaches to the plant. That's not infection yet, that's inoculation. Then we have infection, which is once it penetrates and gets inside and starts causing disease. So it penetrates, there are obviously some host recognition factors it needs to work out, is this a susceptible plant? Or maybe it's highly resistant, and at this point the genetics kick in and stop further invasion. Assuming it's susceptible, it'll then do further invasion of the plant, it starts spreading, starts to colonise, starts to cause disease. At that point, you get reproduction of the pathogen, it's increasing, your bacteria has got into the plant, it's starting to reproduce, it's starting to spread, and it's starting to form symptoms. Symptoms are the expression of the disease. So, where we had before, yellowing, stunting, necrosis, rotten spots, whatever. So presence of symptoms, which are the expression of disease. Some pathogens might have multiple cycles in the one season, so it might produce a secondary inoculum, or a second set of spores, or a second set of something that so your diseased plant starts to sporulate on the surface. Those spores can then spread and start the whole cycle all over again. You can get a second cycle, a third cycle, and a fourth cycle, and get exponential growth of disease in the crop. So you might start with one diseased plant and might end up with the whole field infected because it can multiply exponentially. Eventually, as the crop gets old, the crop dies, you're going to harvest it, you're going to get rid of it. The pathogen has to work out how to survive between crops. So it usually produces over-seasoning rather than over-wintering, because sometimes it needs to survive over a summer period rather than over a winter period. But it needs to find a way of surviving and remain dormant until it produces a secondary lot of until a susceptible host turns up and it can produce inoculum. That over-seasoning stage can be a dormant spore sitting in the soil. It could be sitting in a canker on a tree, just sitting there waiting for the new season to start up, and then it can, the environmental conditions to be conducive and it can start spreading again. It could be jumping it from the crop into an alternate host, into a weed and sitting in that weed. It could be sitting with the seed and just waiting for that seed to germinate and then start the whole infection cycle all over again. So there's a whole range of different strategies that pathogens can use to survive between crops. So that was all I wanted to finish off from that previous thing. So again, in terms of biotic pathogens, their interactions with plant tend to occur in distinct cycles, which reflects the time factors and the interactions of at least two biological entities, the host and the pathogen, and the environment over time. So pathogen develops, produces spores, the plant's developing, you get inoculation, infection, that interaction occurs, disease expression, and away it goes. All of those take time, and all of those will occur at distinct times and distinct processes. There might be environmental drivers of those, there might be plant developmental stage drivers, there might be pathogen drivers. And understanding those are really critical if you want to break that cycle. So if you understand that penetration and host recognition must occur in seedlings because mature plants are resistant, you do your best to protect it by maybe applying fungicides or actually trying to break that cycle at the very early stage, because if you protect it before infection occurs, then when the plant grows up you can ignore it because it won't cause any disease. Or if you can target the dormant stage and actually reduce that in the soil, then you might be able to and understand how it survives. If it's surviving in the seed, for instance, maybe you can certify the seed and make sure that it isn't carrying the pathogen, or maybe you can treat the seed some manner and get rid of it, and then you actually break the cycle. So understanding the pathogen cycle will allow you to develop effective control. Okay, any questions at all about that stuff we've covered so far? Good. I probably couldn't answer them. No. So, next one. We'll start what we're supposed to start now. We're moving on a little bit more about talking about the principles of diagnosis of plant diseases. So what are the processes, what are the best processes for trying to determine what a disease is? And I hope you've already sort of gathered that it's not just get a leaf, take it to the lab, stick it on an agar plate, work out what the fungus is, and that's the answer. That might be part of the process, but it won't tell you the whole story. So, diagnosis aims to determine the cause of the abnormal condition, and more than just IDing the pathogen. Diagnosis obviously is useful for a range of things. It can help you identify that you need to do something, that you've got a problem to deal with. It may also help you understand more about the pathogen, more about the disease itself and how it's spreading. So if you can work out not only what's causing it, you might work out the things behind it, things that are actually dealing with it, that will then be able to develop a decent management control for that. So let's go how we go about examining diseased plants or bits of plants. The first and most obvious one is looking at the symptoms. And we're going to do this in the prac class tomorrow. Is just look at a whole bunch of symptoms. Symptoms are the expression of disease on the host, reflecting those interactions of the pathogen and the environment. So here you've got a necrotic spot. Now that's not the pathogen itself. It's not just the host. It's the interaction of the pathogen and the host, which lead to a bunch of dead cells and leads to this spot that's surrounded by yellows, sort of chlorosis around it, which is probably indicating toxins present. So that is a symptom. For many diseases, characteristic symptoms may be sufficient to actually nail what the actual disease is, and then you can go down and look in the literature and find out all of that background disease cycle stuff, and even what the recommended controls are, and you can actually shortcut the whole process and actually lead to a good outcome in terms of management of the disease. So understanding symptoms can be really valuable. And it might depend on the characteristic symptoms could be related to a specific pathogen causes that specific symptom. But even if it's just understanding that actually it's the roots that are rotting. So I'm probably dealing with something that's actually in the soil, or something that's actually maybe a fungus that's causing the roots. It's probably not a virus that's come in through the foliage, because I'd see a foliar symptom. It's probably not a spore that's landed on a leaf and it's led to root damage. So you start to understand at least what group of pathogens you could be dealing with, and also where to look for it. Symptoms allow you to monitor disease. Not only are they useful to try and work out what's going on, but even once you know what's going on, they allow you to monitor it over time. And they can provide either an early warning system for, you've got a healthy crop, you keep scouting until you eventually see a symptom. You're expecting every year you get this disease at some stage, and you're expecting it to occur, but you keep scouting until you start to see it, and then you know, right, now I need to intervene and need to put on a treatment. It might be, okay, I've got to put on a fungicide spray as soon as I see the first symptom, or as soon as I get more than 5% of plants infected, now's the time to put in that spray to minimise the damage. It can also, even once you've got it, it can help be useful to determine how effective your disease management's been. So you've got the disease, it's starting to spread, you go in there with an intervention. Did it work? Has it slowed? If you monitor the spread, hopefully the spread stopped or slowed. If it's still going like crazy, it might suggest that treatment you applied didn't work very well, and maybe you need to look at something different. So you can monitor the spread of the disease, you can monitor the efficacy of your control strategies, and that can help you make decisions, if not for this season, for subsequent seasons too. But now we're going to have a bunch of pictures. We're going to look at some of the major classes of disease symptoms that you're going to see in plants. And you can break them down into these 4 very general areas. You can have generalised growth effects, stunting, sad looking plants. You can have tissue death, necrosis, as we've mentioned before, so where you've got cells that die and lead to dead cells. You can have discoloration, chlorosis, other colours, and you can have abnormal growth, actually excessive growth in some instances, where you get gall formation, or you get other stimulation of growth that's not beneficial to the plant. We can also get pathogen signs. Who knows what a sign is as opposed to a symptom? Anybody got any ideas? That's okay. I'll leave it. We'll show you what number. Generalised growth effects. So we can talk about hypoplasia, which is reduction in plant development. So essentially, a great many diseases end up reducing the ability of the plant to grow to its genetic potential. So it might be stunting of the whole plant, it might be stunting of a plant part, it might also be abnormal development, it might be twisting, leaf curling, puckering, all sorts of stuff that actually, because the cells aren't growing or dividing as quickly or as well as they should, and you've ended up, or even the necrosis obviously stops development of that particular part of the plant. We've got some pictures in a minute. So you can have generalised reduction in growth, or another one that probably falls under this category is wilting, where you upset the water transport system and all of a sudden the plant stops to, becomes droopy, non-turgid, starts to collapse down and may well die. So, some examples. Distorted growth. These 3 at least are all virus ones, or that one could even be a herbicide one, but anyway. Shoestringing. So here you've got a tomato leaf that's got probably cucumber mosaic virus in it, and it's interesting because all the veins actually have grown normally, but all the lamina has been greatly reduced. That's stopped dividing and stopped developing, and so you've got these pretty ugly looking leaves that are obviously photosynthetically nowhere near as good as a proper leaf. So an infection that's led to reduced growth in a particular tissue type. rugosity. So you've got all these rough looking leaves instead of smooth leaves. Once again, this is probably a viral example where you've got some cells where the cell division is reduced, and so they're dividing really slowly, and some cells where cell division is going like crazy, it's going like normal. And so you get this uneven growth and you get this puckering and unusual shapes. This one here is probably a necrotic infection in the fruit, and so that cell stopped growing, and then all the others are growing around it, and you end up with weird looking shaped fruit that's not terribly marketable. Leaf curling, again, that's probably some cells growing faster than others and leading to distortion of the leaves. Wilting. Here's a bacterial infection of a tomato. Bacterial wilt, a very nasty one, tends to be more in the subtropics or the tropics. But the bacteria invades through the soil, blocks the xylem, and the plant just goes bloop and disappears and dies because it doesn't get any water up through the rest of the plant. Very, very quick death and pretty nasty disease. Here's a fungal example. So Sclerotinia in lettuce. And here they look like they're melted. So the pathogen invades through the crown region on the soil surface, takes out and cuts off, so you've got necrosis, you've got cell death, but you're actually damaging the water flow, and so the rest of the plant, which is 99% water anyway, just collapses down and disappears. So again, very rapid. Here's a seedling disease damping off. So once again, you've got infection, necrosis around the root part of the cotyledon, and stops movement of water through the plant. It just stops and dies. I thought I had a picture of stunting, but anyway, obviously stunting is another one where they just don't grow terribly well. Necrosis, death of plant tissues. So again, quite common with a lot of pathogens where they will kill plant cells. So the pathogen's invading the plant, it's interacting with the plant, and leads to cell death. Now, the cell death can be caused by a whole range of different factors. It might be the pathogen produces a toxin. So the toxin upsets the metabolic processes in the cell, and the cell dies. And that might be important for the pathogen. It might be the pathogen wants to kill the cell breaks open, that releases all this goodies, and the pathogen then starts chomping away at the material that's left behind. They might use enzymes, particularly cell wall degrading enzymes. So a lot of plant cells rather than animal cells or human cells or anything else, one of the big things that differentiates plant cells from everything else is this cell wall. A very thick cellulose, pectin-dominated structure that's quite robust and holds the cell in place. Now, the internal part, you've got the cell membrane. This is often under high turgor pressure where there's moisture in there and everything's nice and happy. There are pathogens, particularly some of the certain bacteria like Pectobacterium that produce lots of pectinase enzymes, and they digest the cell wall and essentially release the cell with its cell membrane. Under high turgor pressure, without this protective structure, it pops open and releases all its goodies, and the bacteria goes yum and feeds on it, and it's happy and the cell's not. So again, enzymes can be really important in terms of digesting a lot of those protective structures in plants. There may be physical stress. It might be with various fungi. They actually produce penetration structures that actually force their way into cells, and in doing so might end up causing cell death. Other ones might actually grow and cause pressure as the colonies develop, and actually lead to death in that way too. There can be biochemical failures, so again, interactions not necessarily driven by toxins, but other interactions with the biochemistry of the plant cell that leads to cell death. And in some instances, and this is a defense process, plants may actually choose to suicide their cells, may actually switch on defense processes that end up killing a few cells, particularly for those pathogens that need living cells to survive. And that will end up with a necrotic spot, but might actually stop the spread of the disease, might stop the spread of the pathogen. So occasionally you will see necrotic spots that are actually a defense process rather than a pathogen-driven process that is actually good for the plant rather than detrimental. Some examples. So here's a nutrient deficiency, zinc deficiency in silver beet, and you've got all this nasty, horrible necrosis. Obviously that's not terribly good for the productivity of your actually that's spinach, isn't it, rather than sorry, spinach. Here you've got a fungal leaf spot on a what looks like an iris to me, or an onion. Actually no, I think it's an onion. So you've got an infection and this necrotic spot which is expanding. If it goes too much further, it'll probably cut off the top of that leaf and actually fall off. But a nice increasing necrotic spot related to the fungal infection. In cereals, a bit like, I mean this is a monocot too, but in cereals you'll often get, rather than round spots, you'll sometimes get these angular spots because the veins go linear rather than angular veins. And so you can get streaking, necrotic streaking. Here's a leaf spotted peanut and a nice round necrotic spot. So you can see that ring of yellow around it. That's probably indicative that there's a toxin involved, so that the pathogen's invaded. It's producing a toxin. That toxin's sort of leaking out and starting to interact with the metabolism of those surrounding cells. They're not happy. One of the first things they usually do is shut down photosynthesis or shut down chlorophyll production. So they go yellow, and then eventually they'll die and go brown. Here's a bacterial leaf spot in pea. And this is an interesting one too, because you've got your necrotic spot, but it's actually surrounded by what looks like a water-soaked, and we call that a water-soaked lesion. So this all looks kind of damp around there. In that instance, the bacteria is killing cells, but it's also punching holes in the surrounding cells and actually pulling moisture out of them, because bacteria needs moisture for reproduction. And so it's actually making the cells around them leaky, usually by toxins or bacterial polysaccharides that actually cause leakage of the cell membranes. And that actually makes this sort of distinctive water soaking type symptom. Here we've got a leaf spot, a bit like that one really, that kind of grows, seems to go around in a target spot, get bigger and bigger and bigger as it spreads. And that can be characteristic. Obviously that's leaf spots. You can get necrosis in other parts, root rots, so damaging through the roots, stem necrosis. In trees, if you get them in the stem, they tend to be called cankers. So if you see that term canker, it tends to be a disease of a woody plant in the stem. Here's fire blight, which is obviously taking out whole stems. Usually that's actually a girdling of that stem, so you've actually got probably a lesion a bit like this in the stem here, and then that cuts off all the vascular system to the rest of the stem and it dies. And you can get necrosis in fruits and things. So another term you may come across is anthracnose. Anthracnose is a term usually to describe fungal necrotic infections of fruits. So here's a poor papaya. It doesn't look very happy with a bunch of necrotic infections. Here's a peach with, again, a necrotic infection that's actually spreading. Here's an onion infected with a bacteria, which is no doubt messy but also very smelly, so just as well we don't have that here. And similarly a tuber infected with a potato tuber infected with a bacterium, that you've got this nasty, horrible blackening and rotting, and probably also a pretty nasty smell. You can get discolorations, as you mentioned before about yellowing and other things. So yellowing is a really common one because one of the major pigments in plants is chlorophyll, which is green, if you start inhibiting chlorophyll production or start upsetting chloroplast formation, you remove the green and you end up with a yellow sort of coloured leaf. So chlorosis or yellowing is production of chlorophyll and is quite common. But you may also get other colours. You might get stress-related colours. So production of anthocyanins, which tend to be red or purple. So you can get reddening, purpling, and even in some instances blackening. It's not cell death, it's not necrosis, but actually production of these pigments in relation to the stress of infection. Examples. Chlorosis. And again, putting up examples here including things that aren't traditionally plant pathogens, but obviously relate to plant damage or of concern. So here's herbicide-induced damage in hops. I think that was an experimental thing that we did, injecting plants with glyphosate for whatever reason. But I think it's glyphosate damage in hops where you've actually got obviously chlorosis, a nutrient deficiency leading to chlorosis and some necrosis in probably, is that grape one? No. A toxin-induced chlorosis. So here's an infection, and you see that yellowing around it even more pronounced than in those previous examples. So this is the toxin being produced that's actually spreading into the surrounding cells and starting to really inhibit chlorophyll synthesis and leading to that chlorosis. Here's something called a phytoplasma, which is a prokaryote, a bacterial-like organism, but produces symptoms much more like viruses, I must admit. And here you've got this unusual growth. So you've got this. These are what the leaves should normally look like. Normal leaves. You've got this witch's broom type effect, and often associated with reduction in chlorophyll. These are all viruses. I like viruses because they're pretty, and they're fun and they're interesting and they cause some interesting diseases. But that's just me. Viruses can induce a whole range of different chlorotic symptoms, and some of these are going to be very characteristic. If you see things like this, you'll go, well, at least I know I'm dealing with a virus because nothing else does that. So here's some typical ones. You can get ring spots. So here you've got these spots or rings of chlorosis where the infections occurred and it slowly spreads and it induces chlorosis as it moves. That partly, all of these light green, dark green interactions are partly related to the effect of the virus and partly related to the defense processes present in the plants. So where the plant's winning, like in a mosaic, where you've got all these light green, dark green spots, all those cells that are dark green, the plant's winning the battle. All those cells that are light green, the virus is winning the battle. And you end up with, rather than complete chlorosis or complete green, you end up with this mosaic. Still obviously diseased and causing a problem, but a bit more diagnostic. Viruses often travel through vascular system, particularly the phloem, so it's quite common to see them associated with veins. So you get this venal chlorosis. So it's moved down here and it's starting to leave the phloem into the surrounding cells and starting to interact in that way and giving you chlorosis. You can get these rings and line patterns and all sorts of weird things. So this is a virus in peanut, which looks really pretty. This is an apple tree affected with apple mosaic virus, and again highlights some of the difficulties potentially of just relying on symptomology for disease diagnosis. This and this is caused by exactly the same pathogen. It's obviously on the same leaf. Here you've got a lovely line pattern. Here you've got venal chlorosis. Two quite distinct symptoms caused by exactly the same pathogen in exactly the same plant in adjacent leaves for whatever reason. Reddening, purpling. So here's a disease called red leaf disease, which understandably produces red leaves on infection. Again, a stress response and anthocyanin accumulation. Carrot red leaf is a viral infection again, where the virus actually affects the vascular transport of nutrients, but also induces a stress response, and obviously you get the reddening in the leaves. Here's a nutrient deficiency leading to yellow reddening. In this instance here, it's not reddening, it's actually loss of reddening. So that's a terrible photo. But tulip breaking virus. So here we've got a tulip that should be red, and it's got all these yellow stripes. So what's actually happened is the virus is inhibiting, in this instance, anthocyanin production in those infected cells, and it stops it producing the red colour, and you just see the yellow underneath. So whether that's yellowing or whether it's just loss of reddening, but it's nothing to do with chlorophyll in that instance. What else can happen? Excuse me. Much less common, you can get excessive growth. These can be nasty diseases, but they're not as common. Usually you stunt your plant or you get reduced growth rather than excessive growth. But what are we meaning by that? Again, the general term is hyperplasia rather than hypoplasia. So an abnormal increase in the rate of growth of the organ or the whole plant. We can have, for example, things like loss of apical dominance. So what does that mean? Most plants will grow, and auxins produced at the growing tip will actually suppress lateral growth. So particularly around this region here, you'll actually have, all these auxiliary buds will remain dormant because it's saying, I want to grow this way, I don't want to spend all my energy turning into a big bush. If you remove that at the top and remove the source of auxin, all of a sudden all these little side shoots start to grow, and you end up with a much bushier plant. There are pathogens that either damage the growing tip or can affect your hormone levels that will affect that apical dominance and lead to that whole bushy process. So it can be altering hormone levels, sometimes, the whole abnormal growth factor can be either altering the natural hormone levels by removing the cell parts that are actually, or damaging the apical meristems, or by changing metabolism in the plant. In some pathogens, they actually produce synthetic hormone analogues, so they're not plant hormones, but they look like them, and they cause abnormal growth in the plant. And in some instances, this excessive growth is actually the plant defending itself. So the plant goes, I'm getting intact here, I'm going to put more cell layers on, I'm going to put a defensive structure up to protect myself against this invasion. It might even be successful, but it might make the product, the fruit or the whatever you're producing look really ugly and actually become problematic for marketing. Some examples. So here's an example of that. So citrus scab. I'm trying to remember, is that a fungus or a bacteria? I can't. I'll ignore that. Infection occurs, and the host responds by producing this superized cells that actually prevent further infection. So it's actually quite successfully stopped further damage. But nobody wants to buy an orange that looks like that. So from the quality aspect, we've got a real problem. From the plant's growth perspective, it might have no yield impact at all. Common scab, potato common scab is much the same. That's a pretty nasty version of it, but usually it's actually just a surface scarring, a surface cracking. And that again is the host, the potato, reacting to a pretty weak pathogen and producing this corky-like, pretty much the same sort of process here, to actually limit the invasion of the pathogen and then stop it penetrating, stop it causing deep lesions like this. It restricts it to small superficial ones, and so doesn't lead to yield loss, but leads to pretty ugly tubers in this instance that no one wants to buy in the supermarket. Crown gall, that's a really interesting one. Here you've got a bacterial infection. The bacteria invades into the woody plant. Well, actually, I'll draw it. Let's do that way. It's more fun. So in this instance, the bacteria, here you've got a root, and it's got a wound site. So it's been damaged somehow. It might be damaged by, well, roots get damaged by all sorts of things. So it's got a damage, and it releases certain chemicals, acetylstringone. I think I've spelled wrong, but anyway. So it's a wound hormone. This compound leaks out into the soil. This pathogen, which is Agrobacterium tumefaciens, smells this stuff, recognises this stuff and goes, oh, there's a wounded root, and it actually swims against the concentration gradient and finds that wound site. Once it finds a wound site, it actually doesn't penetrate inside the plant. It finds, here's a wounded cell, plant cell, whoops, make that more of a plant cell, and the bacteria binds to it, recognises it, binds to it, forms little joints, and actually does what bacteria tend to do. Bacteria, the way they have sex essentially is that they have conjugation. So a bacterial cell and another bacterial cell, and they'll form little pores between them, and then they can swap nucleic acid between cells. This is exactly the same, except in this case you've got a bacterial cell and a plant cell, and it's effectively conjugating with the plant cell. It has what it calls its TI plasmid, or TI DNA, tumour-inducing. So this transfers that DNA into the plant. That DNA integrates into the plant cell chromosome, genetically engineers it. This is nature's genetic engineer. So this is a natural process. You'll get bacterial DNA expressed in the plant gene. That bacterial DNA produces synthetic auxins and synthetic cytokines. They cause that. So once it's engineered, once that DNA is transferred over, those cells can't help themselves. They're constitutively expressing these hormones that are causing cell division. So they divide, and form these massive galls. Even if you kill the bacteria at this stage, that won't stop that, because it's the plant doing it now. The plant is actually engineered and keeping on going. The bacteria, why does it do it? Because it also introduces a gene that actually makes the plant produce a particular amino acid that only it can eat. So it's a kind of a specialised opine, they call it. And this compound is the bacteria's food. So it's essentially turned the plant into a food factory. So you've now got, instead of one cell, you've got a million cells all producing these opines. And the bacteria sits around the outside of the gall and goes, yum, I've done my job, I can just sit here and live and grow and eat and be happy. Plant's not very happy, but that doesn't matter. Anyway, that's the story. Here we've got a nematode attacking a carrot, and once again, invading and causing these giant cells and causing this excessive growth of the carrot. Inside each one of these little galls will be a female nematode that's happily sitting there feeding and laying lots of eggs. But again, it's affected the hormonal processes in the carrot root and caused that cell to expand and divide. We talked before about rhizobia-induced nodules. They are a really great disease symptom, except they're not a disease. So much the same, they actually look very similar to a lot of these ones in terms of bacterial infection, induction of excessive growth. It just so happens that usually it's beneficial for the plant rather than detrimental. You can get some weird things. This is loss of apical dominance. So all of a sudden, like the compound's called a witch's broom, where you get this excessive shoots growing from all the auxiliary buds. Similar thing happening here. So hairy root disease. In this instance, there's a couple of things, and one of them is actually a different version of Agrobacterium that actually induces hairy root and kills apical dominance, and all these little side things start growing, so you end up with a hairy looking carrot. Phylloidy is a really weird symptom. So this is caused by a phytoplasma that we will talk about later, these bacterial-like organisms, but they can, in this instance, cause reversion. So this is a strawberry flower that's turning back into a strawberry leaf. So it's actually changing the actual differentiation of the plant. And this is enations, where you get these weird little outgrowths on leaves. And there's not that common, but a few pathogens that can induce these sorts of outgrowths. Okay. Signs. What's the difference between a sign and a symptom? The sign is actually the pathogen itself. And it's obviously really, really good for diagnostics. So where is the symptom? All of these guys here. This is the plant responding to the pathogen and doing stuff. You know, growing lots of little hairy roots. The pathogen responding and growing galls. That's not a bacteria. That's a gall. Those are the actual pathogen itself. So here we got botrytis, a fruit rot. So this fungus is attacking the raspberries, and it's producing millions and millions of spores, or conidia, and they're greyish coloured. So this disease is called grey rot, and you see that, and you can tell, you see the symptom of the disease, but you also see the sign of the pathogen. You actually physically see the conidia, and so you'd be pretty confident, rather than just a dead raspberry, you're actually seeing grey spores. You'd be pretty confident that you're dealing with botrytis. There's one other disease that does something kind of similar, but you'd be pretty confident of what you're dealing with because you actually see, you certainly know it's a fungus at least because it's covered in fungus. Similarly, here we've got same pathogen but producing a resting structure, a sclerotia. This melanised hard structure that's its survival structure. So often in infected plants, like we're saying, over winter, it needs to survive somehow. So some of these fungi and some of these other pathogens will produce survival structures. So in this instance, a melanised lump of fungus, effectively, that will actually embed itself at the infection site, and then when the plant falls down and starts to rot in the soil, it will be released into the soil. But presence of these sclerotia are obviously pretty good diagnostic tools. You can say, look, I know I'm dealing with a pathogen that produces sclerotia because I can see the things. It's not just dead tissues. Here's a mulberry with sclerotia as well. Not as easy, but you get a few of them here. Here's a post-harvest rot. So again, you've got corn busy rotting in storage, and you see the fungal mycelium. So you know there's a fungus rot there. It's not just dead stuff, it's actually the presence of the fungus as well. And not only that, in this instance, what else is it about that fungus that you can tell? Anyone see the colour? It's a bit pinkish. So that, again, is really characteristic. You know, Fusarium tend to be pink in culture. So here you've got a fungal rot and it's pink, and you go, look, I've got a reasonable idea. It could be that. You go and get some books on post-harvest disease of corns and find out about Fusarium kernel rots, and you go, yep, that's probably it. Then you can go and do a further test just to confirm it. But again, some diagnostic, because you can actually see the pathogen itself. Here's some other really good ones, and these ones are quite common in terms of occurrence, but also as diagnostic tools. The rusts, mildews, and smut. So these are all obligate parasites. None of these ones can be cultured on agar plates. So if you did say, I've got this, I'm going to put it on an agar plate and see what I grow. If you grow anything, it won't be that. It'll be some contaminant, because none of these actually grow on agar plates. But they're pretty obvious in terms of their signs of what they're showing. So here's a rust. You can see these bright yellow spots, and when you get a bit closer, you can actually see it's just covered full of all these spores. So rust spores. Many plants have different rusts. They're all different species, but they're nice and diagnostic. Here's another rust. It's a brassica. It's actually a white rust. So instead of being yellow, most of them are yellow, but this one's a white one. You've also got the mildews. So here's lettuce downy mildew. Downy mildew and powdery mildew are quite distinct fungi. The easiest way to tell them apart is downy mildew tends to occur on the downside of the leaf. Powdery mildew occurs on both the downside and the upside. But once you look down a microscope, you can tell the difference really, really easily. So here's a downy mildew with, again, lots of spores. Here's a powdery mildew in apple fruits. And again, all this white fluffy stuff is just spores. So it's very easy to tell. This smut, that's what a corn kernel should look like. This is a big fat one full of black spores. It's obviously not one you'd want to eat, and it's, again, fairly characteristic of what you're dealing with. You might also have fruiting bodies, little mushrooms. So here's these little apothecia. In this instance, is growing on an agar plate, but these can actually form at the base of a plant, infected plant, or even on the side of an infected plant. All of that steam stuff is actually spores. It's not steam, so that's just billions of spores being released. These pretty interesting looking fungal spore structures that hold spores. They're growing on an infected tree. Here's again an infection that's starting to produce these fungal spore-carrying pycnidia that are structures that again are going to be quite characteristic of the particular pathogen you're dealing with. You might also get extrusions. So here's a rust that's also releasing a whole bunch of sap from the plant, so you end up with this yellow goop. Again, the presence of the pathogen plus some plant stuff. Bacterial cankers will often cause this sort of leakage. If you've seen some fruit trees, you'll often see this sort of sap leakage, generally caused by bacterial canker. There is a fungus that can do something similar, but that's caused by the bacterial infection inside, building pressure, and then it actually forces cracks in the surface, and then under the right conditions will exude sap and stuff. So that's plant sap, but it's also absolutely chock full of bacterial cells. Here's a potato with bacterial red rot. How can you tell? You chop it open, give it a bit of a squeeze, and you see all this ooze coming out from the vascular tissues. Pretty nasty and pretty obvious what you're dealing with. Now, fungus isn't going to ooze out like that. And here's a root under a microscopic examination. You actually see the nematodes sort of stuck to the outside. So these are nematodes that sit on the outside and feed rather than penetrate inside. Again, sign of the pathogen. Really, really clear what you're dealing with in terms of diagnostics. How far have I got to go? Oh, we're going to finish it off next week, that's fine. These pathogen signs, I said that they're really valuable in terms of diagnosis because they tell you what you're dealing with. They can be encouraged too to assist in diagnosis. So, for instance, here we've got grape leaves. They're testing for downy mildew. We saw some downy mildew before on that lettuce that had those nice sporulating structures. Well, sometimes you get an infection, they haven't quite got to the stage of sporulating, or maybe it's been warm and dry outside and so it's not conducive conditions for sporulation. So let's take the leaves, stuff them in a plastic bag, increase the relative humidity and see what happens. And you might encourage sporulation, that you can then say, oh, okay, that lesion I saw has sporulated now and it's got lots of downy mildew spores on it, so I know what I'm dealing with. For some other cultures, you might isolate something and you can't tell what it is. Treat it with ultraviolet light and all of a sudden it starts sporulating, so it can actually be, you can assist its development. Oop. So, I think we might stop after the next slide. So, in sort of summarising it or putting it together, if you're going to use symptoms for diagnosis, first you need to examine a range of your disease plants, because while we've got a whole bunch of symptoms there, they might vary. You might vary with different stages of plant development. They might vary with different strains of the pathogen. You want to see what is the spectra of symptoms you've got. You want to examine all of the plant too. So you want to know not just the leaf spots, but what about the roots? What about all the other parts? What about the other bits that are affected? You want to understand what are the major symptoms, what tissues are affected, and also see whether there's any signs of the pathogen present. So in summary, symptoms are the external manifestation of disease. So they're not the pathogen, they're not the host, but they're the expression of that interaction between the pathogen and the host. A sign is obviously the actual pathogen itself as it's interacting with the host. If you understand symptomology, it will help you with diagnosis of disease and disease management, but it may not be sufficient by itself. Disease is a dynamic interaction, as we discussed before, and symptoms may vary with environment, may vary with variety, may vary with pathogen, may vary with time. So they can be really characteristic, but they may vary. Similarly, you might get multiple organisms causing something very similar. You know, a necrotic leaf spot might be caused by 3 or 4 different things, but at least it starts to narrow down the 3 or 4 things you're looking for. You're not looking for a root-rotting pathogen if you're getting spots and leaves. And we'll park it there till next week. Thank you. Any questions?